We are live, sir.
Okay. So good evening. Welcome to this second live session for the course Human Molecular Genetics. I hope all of you are finding the lectures easy to follow and enjoyable as well, because this is a subject, something that you may not get as a textbook, the topics that are covered here. So in that way, it is something that is a new initiative. I'm sure you are finding it useful as part of your curriculum. If you are a student crediting this course as part of your curriculum, or if you are a working professional, you are finding it as career progression in understanding what are the new approaches, then I'm sure that is of benefit to you. So first, what we will do is that we will take up the questions that are there in the spreadsheet. I think there are some 4 or 5 questions that are given. In the meantime, those who have not filled in any question but are interested in having some discussion, which are not sort of covered in the topic, or you have some allied question that is going beyond the topic that is covered, then I would take it up. You can type them in the chat box as part of your live video. We should be able to take it up. But I would recommend that you please do not ask any question or type any question with regard to the exams or your payment of fee or exam centers and things like that. And this is not something that is handled by the instructor. It is handled by the IIT Madras team. So you may post them in the discussion forum. You'll be able to get a response. And likewise, questions on whether the deadline can be extended, whether assignment 1 is considered. Again, this is not something that is meant for the live session. This is more to do with the topics that is covered. Therefore, I would refrain from answering any such questions here. Okay. Yeah, with that, we will move and take up questions that are written there in the spreadsheet first, and then we'll move to the YouTube link where you can continue to add questions. We'll take them one after the other. Okay. And I'm sure the quite good number of participants have joined already, around from 15, 16. So I'm sure you'll be able to add the questions. There's one question that is, again, there is a question on the assignment. Please, these things that I'm not going to answer because the answers are already given in the dashboard for the particular course. You can go and look at it. It's best of 3, whatever it is. But anyway, I'm taking the technical questions alone. And right now, all 3, there are 3 questions, all of them from Supratik Devnath. Yeah, so the question is that can popular reverse genetic approach conditional knockout using the Kreylock's technique be used to inactivate or silence any particular defective gene in case of in vivo studies using live cancer cells obtained from biopsies and so on? Well, I mean, technically, yes, yes, you can do that. But the answer is like what benefit it is going to give you, right? So, for example, I think you are asking a question: if there is a gene that is overexpressed and leading to cancer, if you do a knockout of that, will it suppress the cancer? But then you take a biopsy and you can simply do an RNAi, right? Much easier than doing a conditional knockout. Conditional knockout you do because you have one cell in which there is a knockout allele. Then you use that because these are stem cells. You can create an embryo and that forms germ cells, and then you can breed them in a way that you are able to create the same mutation in every cell of that organism. So that objective you cannot achieve by doing a similar approach using cancer cells. So that's it's not going to laborious approach. It's not going to help you. You can simply do an RNAi, if you're wanting to study the cells, but not as a therapy, right? So how balanced translocation may affect any individual of or the success generation, how these can be identified properly. Um, balanced translocations are not causing the disease. That's why you're seeing a normal individual having translocation. There is no net gain or loss in the genome. So except that the chromosome broken and joined elsewhere, but you have all the genes that otherwise you have in your genome. Then what would happen in the next generation is that when there is a segregation during meiosis, you don't expect that the two broken parts of the same chromosome, get to the same gamete. So it may or may not. If it doesn't, then that is going to be an issue because that gamete will not have all the genes as part of that genome pack. So obviously, the individuals who are having the balanced translocation are going to be at risk of having such difference in the genome composition in the next generation, right? Yeah, that's very high. In fact, they are at high risk of recurrent abortions and so on. That is well known in the literature. How biomolecules used in identification of coding sequence of any particular oncogene? Well, I mean, this is you name something as oncogene is because you studied and you found that it is causing the cancer. That's why you give that name as oncogene, and that is done by looking at either that region of the chromosome which is having the gene is amplified, meaning several copies of the gene is there, or it is for some reason it is transcribed more transcript than otherwise, or ectopically expressed, meaning it shouldn't be expressed in a given time in a given tissue. Now it is activated. So as a result, you have the cancer. So that's how people have done looked at. It is either the traditional way, if you look at it, people have looked at aneuploidy or translocations associated with the cancer. They looked at the breakpoints and they found, for example, because of the translocation, a gene which should not be expressed now expressed. And that's how they found. I mean, later when you have had these more modern approaches like microarray and RNA-seq, so now you know what are the changes at the level of transcriptional, regulation, and we know many genes that are turned on, which we call as oncogene, or it could be what is called as tumor suppressor gene, which suppress the tumor function, and now this is no longer expressed or it is inactive. So therefore, there's a higher probability of having cancer. So you name them based on their function. Therefore, you have understood the transcript or the protein, and that's how you named it. So it is the other way around. That's where it is. So these are the questions that are there in the spreadsheet. Maybe now we will look at some questions in the chat box, but I don't see any question right now. Is it Murali? Is it Rashmi? Is that only I don't see, or you also don't see any question?
Students are yet to
No.
post.
Yeah, there are no questions.
Okay, good. Yeah, right now there is nothing. So maybe we will wait for another 5-6 minutes. Like, if you could type your questions there, then we will take it up. In the meantime, I also would like to, wish all of you the very best for the exams coming, because the course will end soon. You will be taking up the exam, so you may study well and do well in this exams. So all the best for this exam as well as the other exam that you might take up, as part of your semester and so on. Yeah. So we'll wait for a couple of minutes for the questions, and then we'll take it up. Yeah. Yeah, looks like there aren't any questions. I don't see questions being added there, and but even then, if you have any queries or questions, you are most welcome to post them in the discussion forum. We'll continue to monitor until you give the exams. We'll be able to help you. Yeah, if there are no other questions, then maybe we can end this session. Yeah, there is one question. Vijay Darshini, how does penetrance influence the process of determining pattern of inheritance and less penetrant phenotype distinguished from an autosomal recessive one? Yeah, it's other way around. Basically, you look at the pedigree and then you calculate the penetrance, right? So that's how it is. So it doesn't, influence in the sense that you look at some 3-4 generations and see how many of them are showing the phenotype, and looking at individuals that have the phenotype. Then, for example, the third generation, first generation, third generation is affected, and fourth generation affected, and that means it is autosomal, right? It's basically the affected parent, one of them is affected. So if it affects both sex and transmitted by both sex, then you say it is autosomal. And then, in every generation, if you see, then it is autosomal dominant. Then it is autosomal dominant with penetrance, right? 20%, 80%, 90%, 100%. But the penetrance is pretty low. Like, for example, penetrance is only 20% or 30%. Then you're not going to see it in every generation, right? It would look like a sporadic disease. So any, disorder that is having a penetrance of 60, 70, 80, 90%, only then you can see it in successive generation. At least there may be a few generations skipped, but if you take a large family, then you'll be able to see it in more individuals. Maybe there are few who are carriers not showing that, but it is invariably it is dominant. In recessive, very rarely you have the condition of penetrance. So it is more to do with the dominant phenotype. And by calculating the number of individuals who are most likely to be the carriers, like, for example, in a generation, there is one of the parents is not showing symptoms, but his or her daughter, son showing, the parent showing, that means that this individual should be a carrier. Otherwise, the mutant allele will not go through. So that's how you calculate as to how many individuals likely to have the mutation, and of which how many are showing the phenotype, how many are not. You calculate the penetrance, right? So that's how it is. Yeah. So it is dominant because one mutant allele results in the phenotype. So it is a dominant. So it doesn't come in the recessive category. Recessive mostly are 100% penetrant. Yeah, I think that's the only one question that was there. So maybe if there are no other questions, then we can close the session. Yeah.
Sure, sir. We will end the session, sir. No questions are being posted.
Yeah.
Okay.
Anyway,
sir, thank
all
you
the best to everyone. Do well in your exams and other exams. Yeah, all the best to all of you.
Thank you so much, sir, for your valuable time.
Yeah.
Bye, sir.
Bye.